So I want you to picture a satellite map of a really bustling city at night. You can see the headlights of all the cars zipping around, right? They group up, they stop at intersections.
Oh, yeah, like a time-lapse video.
Exactly, a time lapse. But imagine there's this massive glitch in your visual system. The actual roads, the bridges, the physical infrastructure that dictates where those cars can go, those are completely invisible.
You just see the traffic patterns floating in a void.
Right, you see the traffic, but you have no idea why the traffic is flowing that way. And for decades, cellular biology has actually had exactly this problem when trying to map the nanoscale domains of cell membranes.
It really has. I mean, we could easily see the proteins moving around like those cars, but the lipids, the fats that form the actual cellular infrastructure, they were completely invisible to us.
It's just wild to think about, and that is exactly what our deep dive is about today. We are unpacking this incredibly dense, revolutionary technical report from Nature Cell Biology.
And it introduces a workflow called Lipid-CLEM, right? Which stands for correlative light and electron microscopy.
That's the one. The mission for today's deep dive is to figure out exactly how scientists built this new camera, basically, to photograph individual lipid species at the ultrastructural level.
Yeah, to finally see the roads.
Exactly. And by the end of this, you are going to understand how cells actively sort their own physical infrastructure.
Yeah.
So lipids aren't just passive building blocks holding the cell together.
No, they're so much more dynamic than that.
Yeah.
But to really appreciate how brilliant this Lipid-CLEM workflow is, we first have to talk about why lipids have been so notoriously impossible to film.
Right, the visibility problem.
Yeah. The primary hurdle is rapid dynamics. I mean, lipids are chaotic. They move incredibly fast across the membrane. So to track them historically, standard methodology meant tagging them with something that glows under a microscope.
Like a fluorescent tag.
Exactly, like NBDPC.
But, and correct me if I'm wrong, there's a massive physical flaw with those historical tags. It causes localization artifacts.
That is exactly what happens, yeah.
Because it's like trying to study the natural, stealthy behavior of an undercover spy, but before you observe them, you make them wear this giant, glowing neon sombrero.
That is a fantastic analogy.
Yeah.
Yes. They can't slip through doorways, they bump into crowds, they just get stuck.
So the physical bulk of the tag actually ruins the experiment?
It changes the molecule's behavior entirely. The lipid transport deviates from how a native lipid would act in the wild. Now, historically, you could bypass that movement problem by flash freezing the cell.
Oh, right, cryopreservation.
Yeah, which preserves the physical structure beautifully. But here's the catch. To know which lipid was which, after you froze it, you still had to attach those bulky fluorescent tags to the live cell first.
Ah, so you still ruin the natural behavior before you freeze it.
Exactly. You trade unhindered behavior for structural visibility. It's a lose-lose.
Okay, so how does this new paper solve the glowing neon sombrero problem? Because they use something called a bifunctional diazurin alkene lipid probe.
Right. And let's anchor this to a specific one they used in the study, PC16.0Y.
Okay, PC16.0Y. Break that chemistry down for me.
Well, the chemistry is highly elegant. They just stripped that bulky fluorescent tag off completely. Instead, they synthesized a lipid with two microscopic chemical modifications, a diazurin group and an alkyne group.
Which are tiny.
Incredibly tiny.
Yeah.
When you look at the physical footprint of these two tweaks, the combined effect is basically equivalent to the lipid just having one extra double bond in its tail.
Wow, so chemically, it's practically a native lipid. I think the authors actually call them innocent tracer molecules.
Wait, dude, yeah.
But, if I'm playing the skeptic here, cells have incredibly sensitive alarm systems. If you just swap a synthetic molecule into a live cell membrane, normally calcium alarms go off immediately. How do they prove the cell isn't, freaking out over these probes?
Oh, they ran an exhaustive validation checklist. I mean, a true cellular stress test. First, they looked at metabolism. They use a 4-minute loading pulse to get the probes into the cell. And during that exact window, practically zero probe metabolism occurs. The native lipidome, meaning the cell's existing fat composition, isn't remodeled at all.
So the cell doesn't even try to digest it or kick it out. What about the physical mechanics, though? Does the cell's import machinery still work with these tracers sitting in the membrane?
Flawlessly. They quantify transfer and uptake, or T for short.
And transfer and uptake is like the classic proxy for monitoring receptor-mediated endocytosis, right? Like the cell pulling materials inside.
Exactly. And the TF uptake was completely unchanged. They also looked at clathrin-coated pits, which literally fold the membrane to bring things in. unchanged.
Not even a slight disruption.
The only statistical blip they found was a tiny 8% reduction in structures called caveoli, and that was only when they used a specific sphingomyelin probe, SMY. But in cell biology, an 8% drop is practically negligible.
Okay, but I'm still stuck on those chemical alarms. Did they run specific stress signaling tests?
They definitely did. They tested an array of probes, PC18.1Y, PE18.1Y, and that SMY probe. They aggressively monitored for stress responses at glycerol, which we call DAG.
Great, DAG.
And phosphatidic acid, or PA, and calcium signaling. The results, absolutely zero stress response.
None at all.
None. And just to prove their sensors were actually working, they ran positive controls alongside them using things like ionomycin and PA18.1 bifrol. Those triggered massive, robust, transient spikes in the signaling pathways. But the bifunctional probes were total silence.
A flawless stealth disguise. That's incredible. Okay, let's do a quick mini recap of this first section for you listening.
Great idea.
First, bulky fluorescent tags like NBDPC distort natural lipid behavior, creating localization artifacts. Second, these new bifunctional probes replace that bulk with tiny chemical tweaks, diazurin and alkyne, which match native lipids perfectly.
Right.
And third, rigorous cellular stress tests, checking DAG, calcium signaling, and transfer and uptake prove the cell acts completely normally. So here's a question for you to mull over. If you were an architect trying to understand a massive, complex building, how would knowing the exact real-time location of every structural brick alter your understanding of how that building actually functions?
That's the perfect way to look at it.
Yeah.
Which brings us right to the operational challenge. We have the invisible stealth probe, but how do we actually capture it on microscope camera before it zips across the cell?
Right, because it's still moving incredibly fast. This is where the methodology gets really cool. To help visualize this workflow, let's use a mnemonic device. Think of it as the LCFC algorithm.
LCFC.
Yeah. Load, crosslink, freeze, click. Let's walk through those mechanics, starting with L.
So L is for load. The bifunctional stealth probes have to be inserted into the outer leaflet of a living cell's plasma membrane. To do this gently, they use an alpha-methyl cyclodextrin-mediated lipid exchange.
That is a mouthful, cyclodextrin.
Basically, cyclodextrin is just a microscopic molecular shuttle with a hydrophobic pocket. It safely ferries the fat-soluble stealth probe through the watery environment outside the cell and deposits it right into the membrane. And the chase time here is strictly capped at exactly 4 minutes.
Got it. So that's load. Next, we hit C, which is crosslink. They use UV photocrosslinking to permanently freeze the lipid to its neighboring proteins. But wait, reading through the source material, there was a glaring physical contradiction here.
Oh, the sapphire discs.
Yeah, the cells are cultured on carbon-coated sapphire discs, and carbon famously absorbs UV light. So if the disc is absorbing the UV from below, wouldn't that completely ruin the cross-linking efficiency?
It was a massive physical roadblock.
Yeah.
Standard bottom-up UV illumination was hitting the carbon and just failing completely. So the researchers engineered a physical workaround. They built a custom 365 nanometer UV LED device.
Plus or minus 10 nanometers, right.
Exactly. But instead of shining it from below, they placed it directly into the aqueous medium above the sample.
Oh, that's smart.
Yeah, top-down illumination through the liquid using a spacer ring to guarantee homogeneous light distribution. It bypassed the carbon shadow entirely. With that rig, a 3-second UV pulse was all it took.
3 seconds. That is brilliant engineering. Okay, so load. Crosslink. Next is F, freeze. Immediately after that 3-second pulse, they use high-pressure freezing.
Cryofixation.
And the total elapsed time for both the cross-linking and the freezing combined is roughly 10 seconds.
And the brevity of that 10-second window is what makes this so revolutionary. 10 seconds is fast enough to trap highly transient membrane domains.
Structures that only exist for seconds.
Exactly, or minutes before the cell dissolves them. Once everything is flash frozen, they do a step called freeze substitution. This dehydrates the sample and embeds it in resin. But crucially, this step also acts as a chemical wash.
Oh, it flushes away any residual probes that didn't successfully cross-link to a protein.
You got it.
It's like rinsing the excess dye out of a t-shirt so only the printed design remains. That drastically reduces unspecific background noise. Which brings us to the final C in the LCFC algorithm. Click.
Right. On-section copper-catalyzed click chemistry.
Because the probe is still invisible at this point.
Exactly. It has no glowing tag yet. But because the lipid is now securely cross-linked to the cellular infrastructure, and it's encased in frozen resin, they can safely slice the sample into ultra-thin 100 nanometer sections.
And then they perform the click chemistry.
Yes. They attach a bulky fluorescent tag and azide directly to the alkyne group waiting on the stealth probe. They snap together like molecular Lego blocks.
And the paper notes they actually stain it twice to aggressively boost the signal-to-noise ratio.
They do. And finally, to perfectly overlap the wide-field fluorescence images with the incredibly high-resolution transmission electron microscopy images, the TEM images, they map them using multicolor fiducials called tetraspecs.
Tetraspecs, that gives them perfect spatial overlap. Okay, let's pull all of that together for our next rapid-fire recap.
Go for it.
First, load the lipids using a cyclodextrin shuttle for exactly 4 minutes. Second, cross-link the lipids to neighboring proteins in 3 seconds using that custom top-down UV rig. Third, freeze the whole system via high-pressure cryofixation in under 10 seconds and wash away the excess. And fourth, click chemistry attaches the glowing tags to the sliced resin so you can finally see them.
Perfect summary.
So listener, think about a complex, fast-moving process you manage in your own life. How could a 10-second flash freeze help you see where the hidden bottlenecks are actually occurring?
It really changes your perspective. But we have to talk about the resin itself now. We have the workflow. But capturing a 2D image is one thing. Calculating the actual three-dimensional density of those lipids is a completely different challenge.
Because the physical film we use, the resin block, presents a chemical barrier. You can't calculate 3D density if you only look at the surface of the slice, right?
Right. The click chemistry reagents actually have to penetrate deep into the resin matrix. And the report gives us this really fascinating tale of 2 very different resins. Let's start with HM20.
Okay, HM20, this is an apolar resin, right?
Yes, it is highly hydrophobic. Because of that chemical nature, the polar reagents needed for the click chemistry reaction just cannot penetrate the block.
So the fluorescent signal is stuck on the surface.
Strictly confined to the absolute edge of the section. We're talking about a signal penetration width of just 0.45 micrometers.
Wait, 0.45 micrometers isn't the absolute resolution limit of the imaging setup like 0.35 micrometers?
Exactly. You are literally barely scratching the surface.
So if HM20 blocks the reagents and prevents 3D analysis, why even bother using it?
Because what it lacks in penetration, it completely makes up for in architectural preservation. It is unparalleled for maintaining the physical ultrastructure of the cell. If your goal is precise Z-dimension mapping and keeping the membrane looking pristine, HM20 is the only choice.
Okay, but if our goal is to measure the 3D volume and density of the lipids, we have to switch to the second resin, K11M.
Exactly. K11M is a polar resin, so the click reagents easily seep into and penetrate this matrix. The signal width nearly doubles, jumping to 0.87 micrometers deep.
Oh, wow. So when they take fully stained K11M blocks and cut them into horizontal sections, do the fluorescent signals overlap?
They overlap perfectly from slice to slice. And this allows for full 3D volume staining, which is mathematically required if you want to calculate actual lipid density inside a structure.
That makes sense. I do have a broader methodology question, though.
Sure.
Why rely on click chemistry and fluorescent microscopes at all? If we are ultimately using electron microscopy to see the cellular roads, couldn't we just tag the stealth lipids with highly electron-dense gold beads? They would just show up organically as dark spots on the EM scan?
It's a highly logical alternative. It's
Yeah.
called the nanogold method, and the researchers actually tested exactly that.
Oh, they did? What happened?
They used a biotin picolinolazide combined with 10 nanometer protein, A gold beads, to label the probes. But the problem isn't visibility. The problem is sparsity.
Sparsity, like there weren't enough beads.
The physical beads are just too bulky to achieve dense labeling. When they looked at cellular structures called endosomes, the K11M resin yielded an average of only 11 gold beads per endosome.
11, that's nothing.
And HM20 was even worse. It yielded just 6 beads.
6 beads to map an entire 3D structural domain. That is statistically useless.
It's virtually empty. The nanogold method works fine if you only need a sparse molecular label directly inside the EM image. But the fluorescence methodology, despite having all those extra steps, is infinitely superior for mapping the true dense distribution of these lipids.
All right, let's quickly lock in those resin dynamics for the audience. Number 1, HM20 is an apolar hydrophobic resin. It blocks deep staining, limiting you to the surface, but it perfectly preserves physical architecture. Number 2, K11M is a polar resin. It allows deep reagent penetration, giving you the overlapping signals required to calculate true 3D volume density. And number 3, While tagging lipids with nanogold beads works in theory, it only yields about 6 to 11 beads per structure, which is far too sparse.
Exactly.
So, listener, if you had to choose between seeing the pristine, perfectly preserved surface of a problem or the deeply penetrating, messy 3D volume of it, which perspective would give you better analytical insights?
I love that framing. And that brings us to the culmination of all this methodology. They took this finalized Lipid-CLEM technology and pointed it at a highly debated biological mystery.
The big question, do lipids just flow passively through the cell like water, or are they actively and deliberately sorted, just like proteins?
Right. And to answer this, they focused on the early endosome.
Which you can basically think of as the cell's microscopic post office, right?
Yeah.
Molecular packages come in from the outside, and the endosome routes them to the correct department.
Exactly. So to map the post office's sorting routes, the researchers used two highly validated protein markers. The first is LDL, low-density lipoprotein.
An LDL is a package destined for destruction, correct. It gets routed into specific holding areas called intraluminal vesicles, or ILVs.
Yes, ILVs. The second marker is TF, transferrin. Transferrin is a package that needs to be returned to the cell surface, so it goes to areas called recycling tubules, or RTs.
And they rigorously validated these routes against known structural proteins, right? Like Rab5, Rab7, and Rab11.
They did. So the protein paths are clearly mapped. The ultimate question is, what is the lipid infrastructure doing? Does the floor of the post office sort itself?
So they tracked a highly specific lipid called sphingomyelin, or SM, utilizing that polar K11M resin to get its true 3D density. Did the sphingomyelin just randomly smear across all the compartments?
It emphatically did not. The mathematical distribution here is staggering. The sphingomyelin was heavily enriched in the intraluminal vesicles, the destruction route marked by LDL.
How enriched?
It's concentrated at 3.64 times the normal density.
Well,
Conversely, it was deliberately depleted in the recycling tubules, the return route, at only 0.49 times density.
and what about the main sorting floor?
The boundary membrane, or BM, was slightly depleted at 0.88 times density.
A density of 3.64 in the destruction route isn't just passive drifting. That is active hoarding. The cell is deliberately shoving sphingomyelin into the ILVs. But wait, does it do this for all fats?
That is the exact question of specificity they had to answer next. To test it, they ran a comparative analysis using a metabolically labeled bifunctional palmitic acid.
Because if the cell treats all lipids exactly the same, the numbers should match the sphingomyelin.
Right, but the palmitic acid showed completely different mathematical partitioning. In the boundary membrane, it sat at 0.73. In the ILVs, it was only 2.52. And in the recycling tubules, it was 1.23.
Let me get this straight. The sphingomyelin was mathematically kept out of the recycling tubules, but the palmitic acid was allowed in.
Yes.
Which proves the cellular machinery isn't just sorting generic fat. It is highly specific to the exact type of lipid molecule.
Exactly. And the paper notes they tracked other lipids outside the endosome too. PCY 16.0 and SMY mapped heavily to the plasma membrane and the endosomes.
And PE 18.1Y mapped heavily to the mitochondria, right?
Yes, while PCY 20.4 sort of shared space between them. The conclusion here forces a fundamental shift in our understanding of cell biology. Lipid and protein cargo routes actively diverge at the early endosome.
Lipids are deliberately actively sorted.
They are. They are not merely the passive fluid that proteins swim in. They are dynamic, fiercely managed components of the cellular infrastructure.
That is mind-blowing. Okay, let's do one final rapid-fire recap of these endosome findings. Number 1, researchers mapped the post office of the early endosome using LDL to mark the destruction route and transferrin to mark the recycling route.
Yep.
Number 2, using 3D density calculations, they proved sphingomyelin is heavily concentrated into the destruction route and mathematically kept out of the recycling tubules. And number 3, comparing this to palmitic acid proves the cell actively sorts its structural fats based on their exact chemical identity.
A perfect distillation.
We used to think lipids were just the cardboard boxes holding the important protein machinery. Now we know the boxes are sorting themselves. So what long-held foundational assumption in your own field of work might be completely upside down?
It really makes you question everything. Synthesizing all of this, the true value of this Nature Cell Biology report is the horizon it opens up.
The so what, basically.
Exactly. Lipid-CLEM isn't just a neat trick for one specific lipid. It provides the first generalizable, universally applicable workflow to study practically any lipid within the 3D ultrastructure of biological membranes.
It's the missing analytical key we've been waiting for.
It really is. We can now begin mapping complex architectures we have literally been blind to. Signaling clusters at the plasma membrane, the intricate lipid rings around nuclear pore complexes, the junctions of mitochondrial cristae. This workflow is the first concrete step toward building a true, comprehensive consensus model of biological membranes.
It completely redefines the landscape of cellular research. And as we close out today's deep dive, I want to leave you listening with a provocative thought that builds on what we've just unpacked.
I love these.
Think about this. If our own human cells possess the highly specific machinery to identify, tag, and actively sort microscopic fat molecules at this scale, what happens when a virus enters the system?
Oh, that's a terrifying thought.
Right. What if a virus hijacks that exact lipid sorting machinery to meticulously build its own viral envelope?
Yeah.
Are there completely invisible lipid highways operating inside us right now, actively dictating the balance of health and disease that we are only just finding the map for?
We're just scratching the surface.
The satellite view of the city at night is finally coming into focus. We aren't just watching the cars drift aimlessly anymore. We're finally mapping the roads. Thank you for taking this deep dive with us. Keep questioning the invisible.